So it's 7:02, so let me hit record and we will get started with the second webinar for the 2025 Florida Society for Ethical Ecotourism webinar series. So this is brought on by UFIFAS Extension, Florida Sea, and Florida Sea Grant. So we put on webinars. This is our second year of doing so. Of topics that might interest ecotour operators to provide value for them to, be able to transmit that data to their clients, to be able to get, better quality information for the areas that they're, touring in, and to really, anybody's welcome to these. So if you're a naturalist who wants to learn more about Florida's environment, you're welcome to come as well. So just a brief thing, if you can mute your mics, that would prevent folks from hearing outside noises. If you have any questions, put them in the chat room. If you have, any questions at the end when we get to the questions slide, you can unmute yourself and ask me personally, and I'll have my contact information at the end too. So if there's anything that you think of afterwards, if you view the recording, please feel free to reach out. There's going to be a quick evaluation at the end, which just helps us determine, what we need to cover, what you found interesting, what you could use. So if you could stay around and hit that evaluation afterwards, there'll be a follow-up email, and if you are watching this virtually, there'll be a link in the description too. So quickly, what is the Florida Society for Ethical Ecotourism? It is a group that was established in 1999 in the Southwest Florida area to establish and maintain professional code of ecotourism ethics in order to encourage awareness and stewardship for Florida's natural heritage. So one of the largest drivers of people coming to our state is our natural environments, whether that's the beach, exploring the diversity in the Everglades, and by providing information, being better stewards of the environment, we are sure to, sort of give that gift to future generations to enjoy, as well as, you know, enhancing the economic benefit. So, if we are damaging our environment and you're an eco-tour operator, how many years you might have left if, you're not adhering to best practices. So, this group's great in providing a certification for eco-tour operators, telling them those best practices. So, if you are a person who are looking for a tour, you could look at our list of businesses that have gone through the criteria, or if you're an eco-tour operator yourself and want to join, it's a great way to network and sort of have a lobby to help you, make a decision, to, bounce some ideas off of and learn some stuff about the environment. So this webinar series is brought on by Kate Rose, who's the Charlotte County Sea Grant agent, Carolyn Kovacs, who's in the room with us right now. She's the Volusia and Flagler Sea Grant agent, and myself, who is the Southwest Florida the Collier County Sea Grant agent. So yeah, without further ado, we're going to go into the main meat and body of our talk today, which is fish morphology and ecology. So we're going to be using external characteristics to better understand fish. So an environment that is 800 times denser than air, 50 times more viscous than air, and 95% reduction in oxygen carrying capacity. There is a lot of adaptations that fish had to sort of have to be able to exploit their environment. And if we pay attention to those, we might be able to better exploit them. So, if you're a fisherman, maybe that increases your catch success. If you're a naturalist and you want to bump into these species, maybe some of the information you learned today will help you, come in contact with them more often. If you're an eco-tour operator, this might increase your interpretation skill of, when you have this fish or this marine organism in front of you, by observing the outside, you can, sort of come up with facts and sort of knowledge of where it fits ecologically by observing those. So, one of my favorite quotes here is structure without function is a corpse, and function without structure is a ghost. As a zoologist, this is very powerful. Basically, everything evolved for a reason, and if it didn't evolve for a reason, you could say sexual selection. So that's why maybe those betas are super bright colored, even though they could be picked out by their predators easier, as long as they're able to carry their genetic material one more generation, they did their job there. So in the breakdown, we're going to go over some, mistaken identities, common kind of ones that you might think, hey, how can you tell the difference between this or that fish? But, with me talking about, what I'm looking at, maybe it'll be a little bit more clear. some examples of that. We'll go over characteristics later of the head, eyes, mouth, ears, eyes, mouth, nose, ears. If you talk about otoliths, that's something we'll also talk about. Nares, then body and tail shape. You can tell, how that fish swims in the environment, which might help you determine where you should look for it or, how you should target that. Biological, environmental considerations that might affect, potentially seeing these or catching these things, tips of, how you can use data out there to, increase your success, and then best fishing practices to help maintain the resource, which, fishing is huge. In our state, fishing is the capital of the world, pretty much. Florida is. We have more world records than any place in the world. Just our state has more than like the entire country of Japan and Australia. Probably doesn't help that the International Game Fish Association is in Fort Lauderdale, but we have over 8,000 tidally influenced uh, miles of shoreline, about 1,300 miles if you just do the whole peninsula of Florida. We know we have the Gulf of Mexico, the East Coast, lots of environmental niches, so people come here definitely to fish. So their diversity of fish, form and function. Fish have been around for over 530 million years, and we could thank them for a lot of the favorite characteristics we have today. There's arguments to be made that, our hands might have developed from sort of evolution that happened in the ocean that made its way on land, like the coelacanth and lobed fin fishes that have tetraploid movement that can independently move each fin, and their X-ray looks more like a hand X-ray than it does like a fish fin. Jaws. Before us, there was just a lot of invertebrates and some jawless fishes like hagfish and lampreys, and then the second gill raker became the jaw. So thank fish for jaws over there. And they lived in super diverse habitats. We could find a fish pretty much anywhere in the world, whether it's an Antarctic ocean where fish have evolved, proteins that resist their blood from freezing, to acidic lakes, basic lakes, tropical puddles, the whole ocean. So there's about half of all the current vertebrate species on Earth are fish. There's about 55,000 vertebrate species and about 27 to 29,000 species of fish. That number is sort of whew, because what do we consider a species? A lot of these might be able to hybridize. A lot of these we might be parsing out different species based on genetics, but they might look similar. So that number is pretty fluid. Interestingly enough, there's almost the same amount of saltwater fish species and freshwater fish species because each little pond or river or stream is basically a site where evolution can happen, rather than a body of water like the ocean where there's a lot of interconnection and maybe, one species can be found worldwide like the wahoo. So like I mentioned, because they live in all these, deep water, acidic rivers that are black, they have a lot of cool adaptations that we'll talk about. They come in all shapes and sizes. They could be, 1/3 of an inch, like these small minnows in Indonesia, to some of the largest organisms on Earth, like the whale shark, that could get 40, 50 feet long. It's a great showcase for adaptations and evolution. Like I mentioned, here's that tree, oh, we got jaws. Okay. Cartilaginous fish. There's about 970 cartilaginous fish. This includes your sharks and your rays. This is the teleost fish. We have about, 20,000 species or more. There's only about 10 species of lobe fin fish, which is like your coelacanth and your lungfish, and then about 105 or 108 jawless fish, so like your lampreys and your hagfish. But like I mentioned, there's so many cool little characteristics we can look at with fish. There's, fish that lay eggs, internal eggs, placental connections, fish that breathe air through a modified swim bladder, desiccation, modified fins. Some fish can be dried out and come back, like the lungfish, to make a fluid-filled sac. Modified fins like the remora that has a modified dorsal to suck on a shark, producing light, bioluminescence, sort of glowing under high UV light. Lures, crazy colors, some that you can't even see are there, antifreeze proteins, some are warm-blooded. Some are long-lived, like the Greenland shark that could live hundreds of years. Other ones might dry up in a season. Some fish go Fresh, salt, fresh. Other fish go salt, fresh, salt in terms of their life history characteristics. Sex changes. There's fish that are born female first, then turn into males. There are fish that are born male first, then turn into females, like snook or clownfish. Then there's fish that are both at the same time. There's fish that can make virgin birth called parthenogenesis, like the bonnethead shark. Parasitic males that feed off the female's blood supply, like the uh, anglerfish that they don't see their mates very often. So what better way than carrying them on with you? Electric mussels and more. So when in doubt, check them out. You could learn a lot from cold rating a fish. This is a chimera of a fish. We have a gag grouper, black drum, sheep's head, tomtate, redfish, snook hybrid. But this is a good way of just sort of looking at all the different kind of characteristics that you can really pay attention to. Not, I'm not expect you to learn these now, but just over time through just being observant, interacting with these things, you'll be like, oh, I guess that thing did have whiskers, right? So hopefully in this presentation, you'll learn why they might have that too. It aids in species identification, and that's a really big one because there's a lot of regulations out there to keep fish populations healthy because without those, then we would overfish. I've been to a lot of different countries and states with not as strict regulations, and as a result, their inshore fisheries really suffer. So, folks might have a negative perception sometimes about management and FWC, but they're definitely needed and definitely a great resource for us to look into, even for fish identification resources. By looking at the fish, you can predict what they eat. By looking at the dentition, the position of the mouth, the gape of the mouth, what environment they might live in, what the body shape or tail shape might be. Like something with a lunate tail, which is like a sickle, might not eat on the bottom. But, there's definitely generalists too. Where they live, how they swim, based on, their tail, their body shape, their coloration a little bit. I might give you a lot of information that might be like, okay, so tarpon, so they have an upturned mouth, they're only going to be eating on the top. Not necessarily, like they eat a lot of crabs, so their mouth is upturned, right? But you could also catch one just soaking a bait on the bottom too. So you could always get surprised too, but we'll learn why they might have had that mouth position to begin with. Trends and then do some digging. A lot of times, I get a lot of fish identification questions, and even if I don't know the species of fish, if I get three of these characteristics that are a little bit different than I can see, then I can at least go into the family level of, what this species might be. And instead of thousands of possibilities to look into, it might be maybe a couple dozen, especially just depending on the fish family. So things to consider when searching for a positive ID. We'll go over this quickly. Best DNA, that's something that's the average person doesn't really have able at their disposal. But, in some law enforcement cases, when you have a fillet and you're like, hey, is that a Goliath grouper? Well, DNA evidence would probably be the only way to actually determine that. Who knows in the future that might be more attainable with, folks cloning their pets or getting their own genome sequence. Look for characteristics that don't change when they are alive or dead or shape or size of the fish, like fin type, shape, position. Relative position of fins to each other is a really good indicator. Spine and ray and raker counts. So spines are those hardened structures that you can't bend. You would snap them, which are like the fish's dorsal fin. Rays are like the sort of softer kind of tissue that are on like their pectoral fins that are bendy. And rakers are internal structures in the gills that, worst case scenario, you can use that for a positive ID, but if you do that to a live fish, you're probably going to kill it. So you want to avoid that. Mouth position, that's not going to change with age. Maxilla shape, that's the mouth bones in relative positions. Operculum, that's the gill plate area. And then special characteristics, like if it has barbels, there's not tens of thousands of fish. Maybe actually there's a lot of catfish species, but not in your area that might have barbels, bills, pharyngeal jaws, scutes, keels, suckers, hammers, venom, all sorts of stuff. Okay, characteristics can be body shape and size. Sometimes people will be like, hey, I think I have a world record fish. And I'm like, what are the chances you think you have a world record fish? Because it might be it might be like if you have a 10-pound porgy versus, that might be a really good kind of indicator that it might not be a grass porgy and it might be a jolthead porgy, right? So everything is about an inch or two on a page, but when you see them in person and you have species to compare to in real time, it's a little easier, but it takes time to sort of build that up in your head of references that you have in terms of memories, patterns, depth and capture area. I'll go into in the next slide, a fish that looks similar to the other one, but they might be dozens of miles away in terms of depth that you might occur each one in. Is there a chance that that fish could have swam from deeper water to shallower water? Yes, but what is that chance? Probably less so than it being that other species. If you're a law enforcement person and you're trying to look at, what is other species in the box and their likelihood of catching that other kind of species, teeth, scale type, all sorts of stuff. The worst kind of indication for a positive ID is color and pictures that are artistic representations because, yeah, those are definitely something that somebody drew instead of, a definite picture. But, like I said, people be like, oh, red snapper. Well, there are a lot of snapper that are actually red. So, how would you tell the difference between these two species? One might have more yellow eye and more yellow edges on the fins. Color's not a good indicator, so that might go to size. If that fish is 15 pounds, probably not going to be the silk snapper. And I'll go into what these species are later. But then this is a vermilion snapper, and this is a queen snapper, right? Look very similar in terms of body shape, right? But then you start looking at other kind of stuff that might not change, like this split dorsal position, right? So this thing has streamers. Streamers can get damaged, they can fall off, they're not always there. This dorsal is a little bit split. But if you saw these two, they're very distinct. But from a picture, that might be page 20 and 21. So pretty mistaken identity for somebody who's just starting to fish and say, hey, I might have a queen snapper. I'm fishing in 60 foot of water. Chances are you don't have a queen snapper. You have a vermilion snapper because they live in hundreds of feet of water. So just little things to think about of
Yeah, I
just
wasn't
color
over there.
being not great of an indicator of, what species is what. Then we have, still same kind of color pattern and everything, but hey, this is a black drum and that's a sheep's head. These fish look very similar, but they have different regulations, right? So we need to know which one's which. So if you're looking at these two species off the top, you're like, I don't know. Just I'm done, right? But if you start looking into it a little bit more, you're like, oh, this has a split dorsal. Well, this is a continuous dorsal, right? So that's a black drum. It has a more sloped head. This one's more of like a pointed kind of rounded head, right? This thing has a subterminal mouth. This one has a terminal mouth, meaning it's sort of in the middle of its face, right? So you can determine where that fish eats. This thing has barbels. That thing doesn't have barbels. These have external kind of incisors and molars for crushing. These guys have pharyngeal teeth. So other kind of stuff, fork tail, not fork tail. Just things to keep in mind as we're talking about fish and as you're experiencing these fish of what you can use to be like, oh, that's easy, that's a blank, instead of you having to take a bit to think about it. Another, mistaken identity one that we see often is Cubera versus mangrove snapper. A lot of times, if, fish are very similar in kind, like visual appearance, they might make the regulations the same for the lower size limit, like permit and pompano. Same thing, I believe, with mangrove snapper and Cubera snapper. I think it's 12 inches in federal waters and, 10 inches in state waters. They look very similar, and oftentimes you might say, hey, the Cubera snapper is the bigger one. Well, in this case, the mangrove snapper is the bigger one. But what else are we looking for? More sort of heavier maxilla. That's sort of harder to determine unless you have the other one right next to it. More spaced out teeth, right? This one's a little bit closer together. Rounded anal fin rather than more pointed. And then this is the nail in the coffin is this vomerine tooth patch. This is more of an arrow shape, while this is a W. And, it's not one of these might not poke out, but you might inadvertently, see these after being aware of it for so long that it becomes very evident. But sort of the difficulty of identifying fish. Other one that's really common and really contested that, might have the same minimum size limit, right? But might have different seasons, which gag grouper, I think they have it from September 1st to September 15th in the Gulf this year. Very short season. Black grouper open year round. This picture I'm holding two black grouper. This picture I'm holding a gag grouper and a black grouper. How are you going to tell apart? Well, even newspapers don't really know it for sure too. So this is a black grouper, and it says gag grouper will close on recreational seasons, right? A lot of people might say, "Hey, black grouper have black on them, right? Black dorsal fin and black bottom." Well, big gag grouper do too, and that's a big gag grouper, not a black grouper. This is a black grouper, but you can see that pigmentation on that second dorsal is the same. But then they have other kind of stuff like, hey, this one has a serrated preoperculum, this one's smooth. That's splitting hairs, but one thing you can see very evident is these burnt orange spots are very pronounced on the underside of the fish rather than they're not being here. So, if you had one of these during now you're getting a ticket. If you have one of these now, you're good. So it's very difficult for, enforcement, for anglers, for everybody to make that, but it's up to us to pay attention to that to, determine and save our resources. Sort of who's the pompano over here? He's color not a good indicator. This is a Florida Pompano. This is a Crevel Jack. How are we going to tell the two apart, right? Oh, wow. Well, this one has a bony keel, so there's bones on here, while these are smooth. Once you know that, easy to tell, right? But what other kind of characteristics can we look for? This one has a black dot on their operculum right here. This one doesn't, right? This is a blue runner, also has a black dot. One characteristic for pompanos are these sort of naked spines on the front of the dorsal. So permit, Florida Pompano, and Palmita are all in the Pompano family. They have different regulations though, so you have to tell the difference between these two. And the difference between these two for minimum size limit is exactly the same. So FWC identified that it is hard to tell the difference, and they made that minimum size limit the same. But once they get bigger, this fish can get about 50 pounds. This fish, world record's like probably like 18. So if you get a 20 pounder, chances are you have a permit. We start getting into a little bit more advanced ID. And when in doubt, throw it out. It's not worth the $500 ticket and ruining your experience fishing. You don't really need that fish possibly that bad if you're spending hundreds of dollars in gas to go get this one amberjack because they live offshore. But you have to start knowing, relative positions of that dorsal and anal fin, right? And these fish have regulations and they're closed most of the year. So if somebody tries to grab one of these and say it's a lesser amberjack, if you get the wrong officer, you might be in trouble. And, that's something that we have a real world sort of example of next. So this is a little ID guide that I can actually include in the follow-up email that you'll receive, but sort of going into like what characteristics we look into to get a positive identification for amberjacks. So this is a lesser amberjack, that's a greater amberjack, that's an almaco jack, that's a banded rudderfish, and they're commonly referred to as amberjacks, but most of the time people are referring to this one, the greater amberjack. So look at the relative position of the eye to the maxilla. Once you start getting there, if you're unfamiliar, best thing, just chuck them back in the water, right? Sort of maxilla shape. This is that top jawbone. It's, more fluid than in, mammals that where it's connected to our head, or our top of our skull. And this is sort of a real world application where I sort of use these characteristics that I'm about to talk about to sort of, be a mediary in a law enforcement case where they had a potential misidentification of a fish. So someone was saying that this was a small greater amberjack and wanted to give them a ticket for it. And I said it wasn't definitively a small amberjack because I was drawing lines from the maxilla to the eye and I saw that it was middle over there, right? So it looked more like a banded rudderfish over there. It's a bad picture, so I couldn't make any definitive things. But if you're guilty, it should be with, no doubt to it. This thing right here, this is no keel. This thing looked like it had like a little bit of a raised edge, which these banded rudderfish have, right? So with that, we determined that, they couldn't definitively say that it was greater amberjack, and I believe the captain, got off. But with that, this is a little preview of the knowledge that you're gonna receive from listening to me ramble for the next 20 more minutes or so. So we see this fish here. It's a very common fish in Florida and also, in the south or sort of on the eastern seaboard. You could catch these in Maryland, and the size of the fish there actually are a lot larger than here. Like if you get like a sheep's head in the teens in Maryland, you're probably uh that's average. Well, any sheep's head over like 8 pounds here is, big one. But you can see the broken pattern of this fish, right? So if this fish was living in like a mid-water environment or living in like a light environment, it probably would not have evolved this pattern because why would it help it? It would just stick out. So where would it live if it had these patterns? Probably some complex vertical relief like mangrove roots, pilings, docks, grasses, right? And then you can see the body is actually highly compressed. So oftentimes with a fish that swims long distances, they might be more cylindrical, more slender shape, more rigid actually, which, reduce more rigidness reduces drag better for long distances. You're thinking of mackerel and tuna. So this isn't a mackerel or tuna. So he's probably going to be, interacting with this complex vertical environment. It has a short body, right? So it's probably maybe some sort of prey item too. It's not a predator. You could tell that by looking at its dentition too, which have those incisors and crushers. So not something that grabs and holds, but shears and crushes. Then it has a lot of heavy spines and scales. So if you're a predator, you're not really evolving as much of these defense mechanisms. And if you have these heavy spines and scales, you're probably living around this complex environment that you could bump into, mid-water fish like tuna, mahi, all that kind of stuff. They don't have thick scales on them. You see this tail that is a fork tail, so it sort of uses its environment a little bit more, maybe not sticks on the bottom so much, has a terminal mouth, so it feeds right in front of it, and has those incisors and molars. So by all those characteristics, we know, what kind of habitat it lives in, where it swims in relation to that habitat, the ecological role, definitely not something that's a predator, so you're not going to probably fish for it at nighttime. How it feeds, what kind of food item, probably something with a shell since it has those dentitions. And if you, sort of infer on that, you can know, where I should fish, what kind of flow, what kind of structure, what kind of salinity, to really target these kind of animals in. So we'll start going over, head backwards. And things to consider, not only for positive ID, but just for yourself when you're observing these fish, because by learning these characteristics, if you happen to fish a different country, different state, but you know relatively what that fish, feeds and moves and everything, its ecological niche, and you might be able to just, sort of copy and paste your existing fishing knowledge or your new fishing knowledge from this presentation to be either be able to, like I said, find the organism if you want to look for it or if you want to catch it through hook and line. So, mouth position and gape, that's a big one. So determining how big that mouth can open can determine what it can eat. This is a bad example here because hogfish can open its mouth really big, but they eat sort of crustaceans. But you could tell that by having its crushing jaws in its throat. But its mouth shape is like this because it shovels its face into the substrate. And, you can sort of infer that by looking at its mouth shape. But yeah. Bones and muscles, if it's a short kind of face with, big, heavy teeth, it might be a stronger bite and more crushing, like some of those big snapper species. Types of teeth, we talked about incisors, but there's definitely fish-eating teeth. There's definitely, just sort of sucking down stuff teeth. Rakers, which are like sort of the white portion of a functional unit of a gill. A gill has the red filaments, and on the other side, when it points towards its mouth, are these projections that are cartilaginous, and we'll look at the differences in that. And then hearing and other senses. So the head, look at the mouth position. So, this is a sturgeon. It has an inferior mouth. Where is it going to live and eat? It's eating off the bottom. It has other kind of characteristics like these barbels. And, once we go into sort of the tail shape too, like you're like, okay, this fish isn't going to be like swimming on the top, but they do jump sometimes. But like they're definitely sucking on the bottom. Subterminal, this is a fish like maybe a drum that could, sort of interact with the mid-water, but also, eat maybe crustaceans on the bottom. Terminal, right in front of it. Maybe if it's more terminal, it might be using its eye, so its eye might be a little bit bigger because it's sort of either catching it mid-water column, and then upturned mouth. So that might be like your arowana or your tarpon that, might be eating things that fall from trees or crabs that float on the tide. But these are also like not to say that tarpon won't take something off the bottom. Mouth shape and gape. So, determining how big that mouth opens up can determine what kind of bait and what kind of prey item it has. Snook don't have very big teeth. They have cardiform teeth that are like sandpaper, but their mouth is super dynamic and it'll open really fast to suck in prey like a bass. So, something that's like a larger presentation, a larger bait might be more appetizing for that fish because it evolved to, strike and eat that. Um, but then also, if that mouth moves around very kinetically a lot, it might be more prone to damage. So we have to handle those fish with care because there's a lot of little bones that could potentially break. And even if a fish swims off, it might not live over time. It's up to us to get them in the water as quick as possible, reduce handling time, reducing contact time. Don't want to take the slime coat off of them to make sure they're there for the next angler or for you when you want to, when the season might be open. Pharyngeal jaws, we quickly talked about those. Those are crushing jaws in the throat. Pharyngeal is like pharynx, so in your throat for crushing. Some fish have actually, some fish don't even have crushing ones. Some fish, like the moray eels, have pharyngeal jaws that look like the Alien movie where they shoot out and grab stuff towards them. Fish don't have forks and knives, they have their mouth. So they have to be able to manipulate the environment that way. Like I mentioned, some things will crush like the sheep's head. Some things will cut like barracuda has very sharp teeth, right? So what would you cut? Would you cut shrimp? No, you would probably cut fish, right? Other things will suck down food, right? Um, and then, yeah, gill raker. So this is a fish that's a wrasse, and you can see that very kinetic kind of shape. It's sucking down food, but all those bones are very fine. And to a lesser extent, like a snook might be doing that, like a tenth of a scale. But that's why you don't want to lip a large fish because it could, dislocate that bottom jaw and hurt it. This is a picture of some pharyngeal jaws, which are located in that throat that crush and further manipulate food. So if you have these, are you crushing fish? Probably not. You're crushing oysters, barnacles, crabs, shellfish. So that's what I would use for it. Other things to look for are like these barbels. So why do they have those there? Those are chemo and mechanical receptors. So they're probably living in very murky kind of water, feeling around with their chin, looking for bait and eating them, right? So we oftentimes see fish in aquarium when the water's really clear. You don't want to have like a brown pond and go like, hey, there he is. Like we put these fish in these artificial environments for us to see them, in public displays, but they might be living in some crazy different kind of waters. And then these are gill rakers. So these are little like comb projections that are inside of, this is actually the red part of the fish, the gill. This is the white part. Long ones means they filter feed water. So like mullet have that. You're probably not going to catch one on hook and line, right? They're eating the algal blooms. As they get stubbier, they're probably more of a predator where they're eating like a larger prey item. So it's not ripping these gill rakers or breaking them. They have to just get them past them. And that's some redfish pharyngeal jaws and redfish teeth. Okay, so other head kind of things, we got eyes. So if a fish evolved a lot of kind of effort in its eyes, has large eyes, it's maybe a sight predator or maybe nocturnal. Maybe it's living in very clear water because if it didn't live in clear water, eyesight wouldn't really help them. So if you live in a cave, you probably don't have any eyes, but you might have all these long filaments that if something bumps into you, you have mechanical reception. Nares, that's their nostrils, so they don't have a direct esophageal connection. Like they can't go and like hock a loogie. Like they're just blind sacs, right? So, some fish you can identify better through that. Like sunfish are native species of sort of looking like cichlids. They have two nostrils, while other fish like cichlids actually have one nostril or one nary. taste. Once you get to barbels, like these things on the black drum, it's very easy to distinguish, or whiskers, it might be a catfish, so you can look into that family. Hearing, these are those otoliths, those ear stones in the ear. That's really getting to it. But, if you're doing fish content analysis, like stomach analysis, those are actually calcium carbonate, and the last thing that that fish will dissolve inside a stomach. So sometimes I can open up a fish stomach and see what it eats based on all these little stones that are inside its stomach that have distinct shapes. Fish that utilize the three-dimensional environment and sort of the benthic orientation have larger otoliths, like a red snapper might have one as big as your thumbnail, while fish that are mid-water, like a blue marlin, might have one the size of a grain of rice. But they're very distinct. And then there's a different kind of movement detection from neuromast cells along the lateral line of the fish, or the ampullae, which are the sort of electric detecting organs on the front of the shark's nose and other parts that look like little pimples. Okay, so then we'll look into sort of the backside of animal and really the body. And by looking at the body, you can just determine how it moves. Fin types, there's definitely a lot of different fin types out there that are distinguishing fin position. This is a way that a lot of people will, identify different kind of sharks, because everybody who thinks a large gray shark's a bull shark is not oftentimes right. And it might be, what is the relative position of these pectoral fins versus the dorsal fin? Are they in front? Are they in back? Are they on the same plane? Body shape and color, but color, like I said, is one of the worst kind of indicators of what a fish is, because it could be different if it's alive, dead, male, female, breeding, active, feeding, all sorts of different things can affect color. Um, yeah. But, by learning how a fish swims, you can determine, where where in the water column it lives, your retrieve type, how, what kind of lure you should use. So movement, fish move differently. Some move with like their whole body, like maybe like an eel. Some move with just their back half, like a trunk fish that, are very rigid. Some just move with like their tail. Other ones have medial fins, like the pectoral ones, and some just pectoral only. So there's just a distinguish between paired and unpaired fins. This is a characteristic for identification. So, they might be like, look at the paired fins, blank, blank, blank. So paired fins would be your pectoral fin, your pelvic fins, and that's about it. And then your unpaired fins would be your dorsal, your anal, and your caudal. And these can have just different variations that you can see here. Fixed versus retractable. Fish can tuck in their fins, but sharks are also fish, and they have their fins out all the time. So it's something to look into too. Maybe some cartilaginous fish like sturgeon have their fish out all the fins out all the time. Then aspect ratio. So aspect ratio is actually the square of the fin height divided by the surface area. So pretty much the more sporty and lunate that fin type is, the more kind of sustained, I'm gonna swim forever for the rest of my life kind of fish you'll have, like tuna and mackerel. So those tuna and mackerel are probably not feeding off the bottom. You could tell sort of where a fish swims based on what its tail look like. So I like this example here. It's a fork tail, right? It's very common, but hey, why is that lobe a lot bigger on the bottom? Well, that's a flying fish's fork tail. So its lobe on the bottom is bigger because it's kicking off the top of the waves, right? So that's coming in contact before any other part of its body, right? But then, you know, I think Jaws' anniversary happened. What kind of fin type do sharks a lot of times have? They have the inverse of that, with that top fin being larger than the bottom fin. So what does that say if this says it swims on the surface? It means they're probably cruising around the bottom, playing cleanup crew, eating stuff that's on the bottom. But then there's sharks like mako sharks and great whites that might have this high aspect ratio. So they might be, more of a mid-water column swimmer. So, what swims more mid-water? Tuna, seals, all that kind of stuff, rather than, you might not see a great white shark eating stingrays. Um, And then size, flexibility, and drag. There's different advantages of having these fins. Rounded, you might be more maneuverable. You might be better on just sort of burst speed, those truncate kind of things, but you might not be good for sustained movement, right? So if you're not good for sustained movement, you're probably around structure, not just endlessly swimming the ocean. And then drag, like I mentioned, fish that are sort of constantly moving are pretty rigid to reduce their energy loss from flailing around. Okay, so body, their shape. So there's there's fusiform, normal, elongated, eel-like, flat, short, deep body, compressive form, and I'm sure there's a lot more others. But really, the spectrum goes from like torpedo, eel shape to flatfish, like a flounder or a stingray. So if you're looking at a torpedo fish that might be more rigid, that might be a fish that is mid-water, like I said, where it's moving. You could also tell by the color. If it's reflective, it's probably living mid-water column because it doesn't have to blend into anything. If it's counter shaded with it darker on the top and lighter on the bottom, it's like a great white kind of aspect of if something's looking from the bottom, it blends in. Something looking from the top, it blends in. But you don't really see any bait fish just looking straight up brown. A lot of times these might be silver because they're in mid-water column. Maybe they're just on sand where where that it's advantageous being like a mirror, rather than something that has to blend into, maybe a structure. Fins. You can see that these things don't really have spines on the fins, so it's not really coming in contact, or heavy scales, so it's not really coming in contact with too much structure. It's probably going to be something mid-water column too. It's probably not a prey item too. You look at its teeth, it has those shearing teeth, so it's not going to be cutting crabs in half. You're going to be cutting fish. It has big eyes. It's a visual predator. So mid-water fish. Probably run it fast because this is looks like a fast fish. There you go. Okay, so what can affect the bite? It's called fishing, not catching. But maybe you could look into some of these and actually increase your success. So life stages, if you're fishing in certain environments that only juveniles are in, tailor your bait size to that, or maybe your approach for that. Size of the animal, that sort of goes with the life stages. Spawning, when animals are spawning, a lot of times they're conditioning, getting a lot more fat in their system that build eggs, so they're probably eating a lot more. So it might be better to fish for them that time. Geographic distribution. So, certain times of the year, fish might be more present depending on migration routes. Right now, it's good for tarpon. Other times of the year, it might be bad for tarpon in my area. If you catch a cobia in my area in April, that's anticipated. If you catch one in August, that's like, oh, what was he doing? So you'll start learning about, what's typical in your area too. Prey availability, if there's a lot of prey out there, maybe they're not as interested in your prey, or maybe you have to match that prey that's in the area because that's what they're eating right now. They're programmed to eat. Predators, if there's lots of predators in the area, maybe that fish is stuck to the structure because they don't want to put themselves out on the line and get eaten. So you have to fish towards structure or move spots. Algal blooms can decrease oxygen, internal anatomy, a lot of different things. So environmental conditions can change too. Really super hot kind of times of the day, maybe not the best. A lot of these fish might be kind of crepuscular, sort of feeding early in the morning, later out of the night. Sun and cloud cover, sometimes that might help a little bit. Lunar cycles. If I've heard folks, fishing for fish during full moons at nighttime, but the daytime bite might not be as good. Salinity, rain, all sorts of stuff can affect, a fish's sort of wanting to bite. But this is other kind of things you could sort of take advantage of is like, hey, if you know fish that female snook are protandrous hermaphrodites, they're all born male and they turn female. And then females go to offshore or nearshore reefs and passes to spawn during the summertime. So if you want to catch a trophy-sized large snook, you probably want to focus on the beaches and passes and offshore reefs in the summertime. Just sort of looking into these characteristics of, the life history. This is sort of a chart that shows dissolved oxygen over the day. A lot of people, think that the lowest dissolved oxygen might be, the start of the night, but it's actually at the end of the night when there hasn't been photosynthesis taken on. So a lot of primary production, like a lot of algae and plants are producing oxygen in the water, and midday might be the highest amount of oxygen. So that might be an influence of them, wanting to eat or maybe what's available in the water. Tips and tricks, look at this comparative biology, like I mentioned, if you wa- are trying to target a fish this shape in Asia, you probably could use similar conditions that you caught them in here to catch that fish, even though it's a different species. There's stock assessments and CDARs. So stock assessments are a great way to learn more about the biology of those fish, sort of, how big do they get till they spawn, where they might be when they do so. All sorts of really great information, and it's free, and that's sort of what a lot of these decisions are based on for management. Scientific papers are great. Commercial catch data is also good. This stuff is all available on the FWC website, and you can sort of look at month, pounds, average price, area of a certain species in a certain county between certain years. So I pulled this up for a fishing club in our area, and it looks like pompano are caught mostly in March, February and March. So if I'm actually trying to target pompano, I'm probably going to do what the commercial fishermen are doing and, fish around that time because they probably know best for that species. Environmental data, you can look at a lot of weather buoys out there now and your own catch data too. So you could look at, your date on your fishing pictures and see when you caught that, what time of day. And, if you happen to get another Kubera snapper in April, like, oh, okay, April might be a good time of year in your area to do that. And then maps. So you can go on Google Earth and try to find deeper water areas or shallow water areas. It's really crappy to go fishing and cast in one area a whole bunch of times and then learn that you're fishing in like 3 inches of water. So, finding aerial views of deep water that moves might, increase your catch success there. And it's called catfishing, not catching. So, like I mentioned, it's up to us to do our part. And, if you don't do all of these best practices, if you do some of them, they're better than none. So, what really increases the survivability of the fish is to get them back in the water as quick as possible with as little handling as possible. So, if you're not going to keep fish, if it's not a fish that you're going to take a picture with because it's not a personal best, and you have a de-hooking device, maybe it's best to just pop him off on the side of the boat so he doesn't, even touch the deck and wipe that slime coat off. That's just another form of immunity, like a layer of skin, and increase, do that time. So, understanding what you want to do when you're fishing, whether that's, teaching, whether that's, targeting a certain species, maybe you're targeting one for dinner, having that criteria of like, if it's this big, I'm going to keep it, in mind. So you're not thinking about it when the fish is flopping around. If you keep on catching undersized or oversized fish or out of season fish, maybe it's, time to move locations because there is a degree of release mortality. Maybe, certain species are worse than others, but let's say 15% of fish don't make it after you catch them. You want to reduce that if it's like, oh, another mangrove snapper that's 8 inches. Oh, another mangrove snapper that is 8 inches. Reduce your contact time, like I mentioned. Hold the fish correctly. A lot of folks will hold a big bass by the lower jaw and just, sort of detaching its dentiary that, affects its ability to open his mouth big to eat real fast. So support the fish's weight. Don't put them on hot surfaces. Use circle hooks. Cut the line if you're gut hooked. Whole bunch of different things. So with this, if you like this kind of education, we have the Florida Friendly Angler program. It's now in Spanish too. It's free. It's online. It's about an hour and a half long that covers environmental ethics, best catch and release practices, and how regulations are made. So feel free to take it if you Google or just type in flfriendlyangler.com. Feel free to just sign up if you like it. Awesome. Go through the whole course. If not, no, it's free. No harm, no foul. But you could just hit play and listen to it. Don't even have to look at the screen. So, gotten great reviews and have about 4,500 people who have taken the class. Okay, so with that, we're up to the questions. I see some the chat has a little bit of stuff going on. This is the QR code for the link, and Carolyn can put the Qualtrics link in the chat if you don't know how to use a QR code. If you're listening to the video recording online, because this will be on YouTube, please, look into the description for that link. But this is also a QR code for myself with my programs that has a little bit more resources with Florida Sea Grant. And that is my contact information. The next webinar will be August 13th at 7 p.m. like today. And it'll be about seabirds with FWC biologist Taylor Ozimek. So I am going to attend that one because I need to learn more about birds. But with that, I will actually stop the recording and take any questions that you guys have. Thanks for listening.